All right, good morning, good afternoon, good evening. Whenever you're watching this is Dr. Kaz, and I am doing our Chapter 6 review. I know it's been a little while since I've actually put up any videos here, so I figured what better time than Sunday. So I've been thinking about it, and I think for the future reviews that I'm going to be doing, I might just use some more detailed recordings from some of my other lecture sections. But that being said, we can I I can discuss that more later. But I wanted to show you folks something real quick here. You do have quite a few. Um, whoops! You do have quite a few resources for this course, and so what I want to do is, all right, boom. Okay, so I want to show you something here that some of you may have been using, some have not. I know there's a lot of material for this course. So in that regard, I want to make sure that what's available to you when you're taking the class and studying the material, there's several different resources. But one of the resources that I really wanted to show you is if we go chapter by chapter, in each chapter folder here, um, you'll obviously start off with the chapter overview and the learning outcomes, and those are important because where we get our test questions for the test come from these lecture learning outcomes, the lab learning outcomes. All right. So obviously, I know you're already familiar with these. These are called the soft chalk lessons. All right. Obviously, the assignments. But what some of you may not have been taking advantage of are the last three folders here in this chapter. You've got your supplemental lecture and lab review materials, and these mock exams are always very helpful. That's what this is up here at the top. That's a mock exam that helps you to get prepared for the lab test. And then we also have some practice activities that are on Connect. Now, again, they're practice, so they're not counted as your grade. So, feel free to actually, if you want to challenge yourself or really kind of give yourself an evaluation as to, all right, am I really understanding this material, this chapter, you can practice your knowledge in these two activities. Now, going back one here, okay, there's these last two folders here. And again, if you have the extra time and you just kind of want to take a little different perspective and on learning the material, you can use these instructional videos for both the lab and the lecture. Again, this is if you're feeling pretty good, if you have time, obviously use these materials. But again, I just wanted to point that out to you just so you kind of had an idea of what is available to you. All right, let's get into shut that down. This is going to annoy me. Hold on one second. I'm going to make this disappear. Awful, awful. Ain't nobody want to see that. Okay. So let's start off chapter 6, the integument, skin. All right, so the couple things that we want to discuss. Obviously, you probably heard this fact before, but the skin is the largest organ in your body. Okay, and there's another name for it. We also call it well, in this chapter, it's the integument system. All right, but the cutaneous membrane. There's 4 membranes that your body has, and you're going to be discussing them this semester. We learned about the serous membrane. Okay, that was that not connective tissue, excuse me, that epithelial tissue that lines body cavities and the organs in some of these body cavities. Okay, then you've got a mucous membrane, and then you got a synovial membrane. We'll talk more about the synovial membrane in chapter 9, all right, when we talk about synovial joints. But in this chapter, we're going to be talking about the skin and then all the other tissues that are associated with it, what we call accessory tissues. So like the epidermal derivatives like nails and hair, all right, sweat glands, these sebaceous glands. We're going to learn about all those in this chapter. Big function of our skin is that it's for protection, but it offers that protection as a barrier around the other organs and tissues of our body, and it keeps you could say it keeps the bad stuff out and the good stuff in. So as you know, there's lots of viruses and bacteria and pathogens that are constantly trying to invade our body, and our skin is one of our first lines of defense. Discuss that when you do the immune chapter in 211, how the skin is part of what we call our innate immune system. Okay, one of the things that I always do, and that pretty much all healthcare professionals do, is that they use the skin as a visual indicator of health. How many times are you able to tell somebody, if their skin has a yellowish tint, you're thinking jaundice, and some of the causes for jaundice is liver damage. But basically what that is that sign that you're able to detect, when you're looking at somebody's skin, can kind of give you an idea, especially if they're coming to your office, and as a medical professional, they're coming to your office and they want to be treated, and you have to figure out what's wrong with them. And a lot of times the skin offers just another tool to figure out what's happening to these patients. One of the hardest specialties to get into in medicine is dermatology, and dermatology is the study of the skin. For those of you that are thinking about going into med school, when you take your STEP exams, it used to be, it could be higher now, but anywhere between 200 and 280. You want to get as high as possible, obviously. Those scores are what you would need to even be considered for dermatology. All right, a couple of functions here. We just talked about it. Protection, physical barrier, helps to keep out tons of pathogens, ultraviolet radiation, any other harmful substances. Part of the function of our skin is thermal regulation, so the skin actually will help to adjust to extreme temperature changes. A big function too for the skin is prevention of water loss and even gain, because we talk about the skin is water resistant, it's not waterproof, and that's evidenced when you lay in the tub for an hour and you get out and your skin's wrinkly. All right. Not only does it help to keep the water in, it can help to repel and keep the water out. Okay. I'm trying to remember if it's in this chapter or if it's chapter 7, in which we'll discuss sunlight, the effects of sunlight on vitamin D synthesis. Vitamin D is a prohormone, also considered to be a fat-soluble vitamin, but it is a substance, we'll just say, that is meant to help with calcium regulations. Calcium levels start to get low, all right, vitamin D plays a crucial role, all right, in maintaining blood calcium levels, helps to the kidneys to reabsorb calcium so you don't excrete it out. And so that's a very important vitamin or prohormone that we need. A couple other functions here, secretion and absorption. All right, obviously what goes in must come out. So some of the waste products for our cells, for our body, sweat, for example, there's a diluted form of urea, in our sweat. So, and a big product of that is nitrogen, and nitrogen is a waste product for a lot of cells. We also use the skin to help absorb certain substances. I'm sure you've seen the nicotine patches. We know that the skin will let certain substances in. It's a selectively permeable membrane there. So it's a great way to administer drugs. Fentanyl patches. I have a dog. Well, it's not mine right now. My father has stolen him from me, but he had back surgery long time ago, and they put a fentanyl patch on him. And it was a great way for them to, for the pet to get that pain relief at a slow, absorbable rate over time. Okay. Immune function, it's huge. One of the most important functions of the skin is its immune function. I mean, like I said, it's part of the innate immune system, and just in general that it forms that protective barrier. But also, when we were talking about tissues, and especially when we were talking about the layers of the skin, we have the epidermal, the Langerhans cells. All right, those are the dendritic cells, and their job is to pretty much just monitor the cells around them and also if there's any pathogens close by. And they'll either destroy the pathogen or initiate the immune response to that pathogen to keep you safe. All right, temperature regulation, that's back in chapter 1 when we talked about thermal regulation with an increase and decrease of body temperature. Okay, so when we want to increase body temperature, the blood vessels in your skin undergo vasoconstriction, and they reduce the blood flow to the skin and keep the blood more towards the internal portions of your body, and therefore it prevents heat from leaving your skin, because your blood is about 1 degree higher than your normal body temperature. So just think, wherever the blood goes, that's where the heat's going to go. So if we keep the blood away from the skin and keep it more towards the internal structures, we keep it warmer. If you're hot and we need to release some of that heat, then we have the phenomenon of vasodilation, and the blood vessels in your skin vasodilate, causing more blood to go to those areas, and the heat follows it, and then the heat will be given off. And then finally, the last function is sensory reception. We have many, many different types of sensory receptors throughout our skin: deep touch, vibration, pain receptors, thermoreceptors. We have tons of receptors all throughout our skin, all right, to detect those specific stimuli, and then we can initiate our response to that stimuli. All right, so let's move into the parts of the skin, okay? All right, so there's 2 layers to the skin. First layer, the epidermis, that's made up of epithelial tissue, specifically stratified squamous epithelial tissue. And so one of the characteristics that we know about epithelium, well, there's many, but one of the key ones is that epithelium is avascular. So your epidermis is avascular. You do not have any blood going to your epidermis, okay, or blood supply, I should say. So one of the things that is a great question that I could ask you is, if you were to get a paper cut and you don't bleed, how far did that cut go into your integument? Okay, and you would know, since there's no blood drawn, then we didn't get past the epidermis. Okay, second layer is the dermis, the deeper layer. Okay, and this layer is the layer that I tell people that has all the stuff in it, and we'll discuss that. Okay, about what's in there in that layer, because it has it has connective tissue, it has epithelial tissue, it has muscle tissue, it has nervous tissue. All right, so the dermis has a lot of stuff. Primarily, the dermis is made up of dense irregular connective tissue, and dense irregular connective tissue does have a decent amount of vascularity involved. Okay, so whereas the epithelium, all right, is avascular, okay, the dermis will have a significant amount of vascularity or blood vessels present there. Now we throw this other layer here into this discussion, the subcutaneous layer, but that is not part of the integument. So you really need to put a star or write this out in bold letters. All right, it is not part of the integument. Okay, but we just talk about it because it's in close proximity to the integument. All right, it's the layer that will separate the skin from the underlying skeletal muscle. Okay, it's primarily made up of fat or adipose connective tissue and another loose connective tissue, areolar connective tissue. So if there's areolar connective tissue there, okay, that means that it's a loose connective tissue, and that means you got a decent amount of blood supply going there. So let's talk about the epidermis here, okay? And the epidermis is made up of 4 to 5 strata, all right, or smaller layers, okay? So you want to know the names for all these layers. So the first layer that we're going to talk about, we're going to start with the deepest layer, okay, in the epidermis, and then we're going to move towards the surface of your skin, or we're going to move superficially. Now the first layer is the stratum basale. Another name for it, and you can be asked this, all right, is the stratum germinativum or the stratum basal, all right? So the stratum basale, okay, is the deepest layer. And right below that, remember, when we talk about epithelium, that epithelium sits on top of a basement membrane. So this layer is adjacent or immediate to the basement membrane, okay? So it's one single cell layer, all right? But look at the description here, okay? Stratum basale, these cells are what we call cuboidal or low columnar cells. But yet, on the previous slide, all right, we said that the epidermis is made up of stratified squamous epithelium, okay? But wait a minute, Dr. Kaj, you're telling me that in the stratum basale, those epithelial cells are cuboidal to low columnar. So that's not stratified squamous. That's right. When we're dealing with stratified epithelium, this is back to chapter 5. When you're dealing with stratified epithelium, when you're naming that stratified epithelium, remember we name it depending on how many layers. So epithelium gets named, if it's one layer, it's simple. If it's many layers, it's stratified. And then we also name it by the cell shape. But here's the thing. When dealing with stratified epithelium, we name it, all right, for the shape of the cells in the apical layer, the topmost layer, the superficial layer, okay? So even though these cells are cuboidal to low columnar in this deep layer, the stratum basale, don't worry about it. As they migrate and move towards the surface, they actually take on the eventual shape of squamous cells. So there's 3 types of cells that we find in this layer. The most abundant and the ones that we'll talk about the most are the keratinocytes. And it's because they produce this protective protein called keratin. The second cell type, all right, are the melanocytes, okay? And their job is to make and store melanin, and that's that pigment that gives us our skin tone. For some of us, if you're me, I have none, all right? I'm as pale as they come. But it's that melanin that is a protective substance for the DNA. We'll talk about that, okay? And what I mean by that is, all right, the melanocytes make the melanin, and what that melanin is used for, when ultraviolet radiation, all right, strikes the cell, okay, melanin gets produced, or the stored melanin gets released, and it goes and covers the nucleus of the cell. It literally coats the nucleus because inside is the DNA. Because ultraviolet radiation can unfortunately splice. It's called dimerization. You can splice your DNA. And if it starts splicing your DNA, you can start to develop some genetic mutations. And that's not good. So your body made melanin to deal with that. The third type of cell are the tactile cells. We also call them Merkel cells. All right? And so they're touch sensitive. And what will happen is when they're compressed, all right, they release a chemical, a neurotransmitter, that will then stimulate some nearby receptors, all right, and inform the nervous system as to what type of sensation, touch obviously, that is undergoing in that area of the epidermis. All right, so those are your 3 cells. Okay, the next layer up, all right, is going to be the stratum spinosum. So the stratum basale layer, okay, that layer, those cells are constantly dividing, all right? And what will happen is, all right, one cell will divide, and then you will wind up with 2 cells. One of those cells will stay in the stratum basale layer. The other one, what we call a daughter cell, will migrate superficially and move towards the surface of the skin. So it'll move into this layer, the stratum spinosum. Okay, so this layer has several cell layers to it. All right, so as soon as one of the cells has divided, okay, then one will stay in the stratum basale, and the other one will migrate into this layer here. Okay, so these cells stop. All right, they're no longer dividing. Now what they're doing is they're starting to form junctions, attachments to their neighboring cells. Okay, because remember, epithelium is tightly or densely packed tissue. Okay, but it's hard for things to get in between those cells, and the reason why is these desmosomes, all right, these intracellular junctions are starting to form. All right, so we don't allow pathogens, bacteria, viruses to squeeze in between the cells. We keep them out. But also in this layer, we find our epidermal dendritic cells, the Langerhans cells. These are those immune cells. all right? And they do a couple different things. We'll learn more about it in 211, all right? But they actually will respond to pathogens through phagocytosis, which is essentially the Pac-Man cell, all right? It'll find something and gobble it up, okay? It's always monitoring the cells nearby, especially because those cells are constantly exposed to ultraviolet radiation, making sure there's no mutations going on. If there's any mutations going on, then these dendritic cells will kill that cell. They're also what we call APC cells. These are called antigen-presenting cells. They can initiate the immune response. The third layer that we see, it's not a very thick layer. It's only 3 to 5 layers of keratinocytes. This is where the keratinocytes start the process of keratinization. And I want you to remember this phrase, keratin kills. Like there was that phrase, crack kills. Well, keratin kills. Because what happens is when the cell produces keratin, it's going to kill itself. That keratin fills up the inside of the cell, all right? It destroys the organelles and eventually the nucleus. And without all that, you can't have any metabolic function, so the cell dies. Okay, but the good is that these cells are filled up with keratin, and this keratin is this strong protein that helps to sustain the cell, all right, and give it support and structure. This fourth layer here, all right, again, another thin layer, 2 to 3 cell layers thick, all right, but this is only found, and you want to make a note of this, this cell layer is only found in the thick skin area. Okay, so the palms of your hands and the soles of your feet. Palms of your hands, soles of your feet, all right, and it's only found in thick skin. So thin skin has 4 cell layers. Thick skin, excuse me, let me rephrase that. Thin skin has 4 epidermal layers. Thick skin has 5 epidermal layers. Okay, so another thing that we note in this layer is during the keratinization process, all right, the cells are producing this elidin, all right, and it's just an intermediate product, all right, during that keratinization process there. And then finally, the outermost layer, the layer that you are looking at when you're looking at somebody else, that's the stratum corneum, okay? 20 to 30 layers thick, so it's quite thick, all right? All these cells are keratinized, which means all those cells are dead. All right, but even though they're dead, they're still affecting a function for the skin, okay? So this outer layer, all right, is a somewhat hostile environment to microorganisms. It's dry, okay? It's thick, okay? So it makes it tough for penetration of these organisms, all right? It offers us protection, like if you walk by something, okay? If you brush up against something, that abrasion there, it helps to protect against abrasion. But the big thing is that it just makes it just a tough area for microorganisms in general to thrive on. All right. All the cells in this area should also be anucleated. They'll have no nucleus because they're dead. And it takes roughly about a month for a cell to migrate to the top of or the superficial portion of your skin and slough off. So what I mean by that, it takes about 2 weeks for a cell to migrate from the stratum basale into the stratum corneum. Then it spends about 2 weeks in the stratum corneum before it eventually gets shed off or sloughed off. Okay, so it's about a month. So it's almost like you're like a snake. All right. Okay. A couple things about the epidermis before we move on to the dermis is you've probably noticed that there's a thickness of the skin, okay? In certain areas, it varies. So, like I said before, with the stratum lucidum, that's found in the palms of your hands and the soles of your feet, all right? You want to understand a couple differences between thick and thin skin, all right? One, you need to know where it is. I think we figured out where the thick skin is. All right, it's also going to have all 5 of those layers there, all right, of the strata, okay? But, and I know we all know this, and you don't even have to memorize this because you just know from living, all right, that you have sweat glands on your feet and on your hands. We've all had sweaty palms at one point, all right? But we also notice that there's no hair follicles, and you can look at your hands right now. You shouldn't have any hair on the palms of your hands. You shouldn't have any hair on the soles of your feet, okay? And so if there's no hair follicles, by default, we're not going to have sebaceous glands, because sebaceous glands are attached onto hair follicles, okay? So no hair follicles means no sebaceous glands. All right, so when we get the thin skin, and this is the majority of the skin that covers your body, all right, like on your face, your arms, your legs, okay? Here, all right, the thin skin, we're not going to have the stratum lucidum. We're still going to have sweat glands, but guess what? We're also going to have hair follicles and sebaceous glands. And if you don't believe me, look at the back of your arm, okay? You have hair there. And if you have a hair follicle there, that means you have a sebaceous gland, okay? All right, let's talk about the second layer here. All right, that's the dermis. Dermis is the deeper of the two layers, all right? And it's going to be the one, like I said, that has all the stuff in it. All right, it's primarily made up of connective tissue proper. All right, in connective tissue proper, there's two types of connective tissue proper. There's loose connective tissue and dense connective tissue. Just a quick review. Okay, so what does that mean? Then that means that this type of connective tissue, or excuse me, this layer, all right, is going to have, all right, a pretty wide variety of stuff in there. Okay, so before you heard me mention, all right, there's blood vessels. We have sweat glands, sebaceous glands, hair follicles, our nails, all right, or nail roots. We have different types of sensory organs in there, and we have smooth muscle. So like I said, the dermis has all four tissue types, all right? It's the layer that has all the stuff in it, okay? So we break it down into two layers, the papillary layer and the reticular layer. Okay, the papillary layer is the more superficial. That's the layer that's immediately adjacent to our epidermis. And then the reticular layer is the deeper of the two layers. In the papillary layer, we're going to find primarily areolar connective tissue. So I want you to keep in mind, when we're dealing with areolar connective tissue, which is a loose connective tissue, and when we have a loose connective tissue, that means we have space, all right? That means we have space for blood vessels. And that's what we're going to see. We're going to see lots of blood vessels in this area, all right? The papillary layer also contains the dermal papillae. And those are these bumps, all right, that we find, all right, just deep to the epidermis. Okay, and in the dermal papillae here, we'll have sensory receptors, like we'll have the Merkel's corpuscle, we'll have the papillary plexus, which are just these blood vessels that will be up in here, okay? And these dermal papillae create this locking mechanism between the epidermis, specifically the epidermal ridges. So the epidermal ridges are these invaginations of epidermis down into the dermis. And it's these epidermal ridges and these dermal papillae that when they interlock, all right, they create what we call friction ridges. But you've seen it on TV known as fingerprints. So when you look at your fingertips there and you can see those ridges there, those are created from the dermal papillae and how they interlock with the epidermal ridges. And they create your fingerprints. All right, so the other layer that's deep to the papillary layer is the reticular layer. This is the bigger one. All right, this is going to have a bunch of the other tissues that we'll talk about in a little bit. All right, but it's primarily made up of a dense irregular connective tissue. And that's great because dense irregular connective tissue is a connective tissue that can actually withstand multi-directional stresses. So you can prove it to yourself by grabbing onto the skin on the back of your hand ever so gently and lifting it off the back of the hand a little bit and moving it around in a different direction. And it can withstand that because of that dense irregular connective tissue there. All right, the final layer that we're going to talk about is the subcutaneous layer. You also want to know its names. All right, hypodermis and the superficial fascia. All right, again, I'm circling this to remind you how important this is, that this layer is not part of the integument. That's a great true-false question. Don't be fooled. It is not part of the integument. All right. It's primarily made up of loose connective tissue, areolar connective tissue, all right, and adipose connective tissue. All right. So if it's made up of adipose connective tissue partially, all right, what are some of the functions of adipose? Because those functions will kind of defer, all right, to the functions of the subcutaneous layer. Well, adipose tissue is great for energy storage because we're storing lipids. What does fat do for us in the wintertime? Helps to keep us warm, so it works for insulation. And then what we'll do is we'll take some of that fat and we'll stick it around some of the organs so that it don't get slammed around, all right, inside of our body. Well, that's awesome too. It offers protection, okay, because of its cushioning. Well, areolar and adipose tissue are loose connective tissue, so therefore this area has a high amount of vascularity, which makes it an awesome place to inject drugs into. Because we know that those drugs, because of all the blood vessels, will be rapidly absorbed and then circulated around through the body. So it's a great place to stick people with drugs. Cool stuff. All right. I'm going to cover nails and then I will stop this video, and I don't want to overwhelm you with too much, okay? And then I'll finish it up in a day or two, or even maybe later on tonight, so you have something more to watch. So I'm going to be going through the nails here. I'm going to be skipping a slide, coming back, and then I'm going to be bouncing back and forth between the two slides here. Okay, so what are nails? And this is every chapter, every system that we talk about. There's always some really interesting, really cool facts that I like to point out. And one of my favorite facts for this chapter is that nails are a modification of your stratum corneum. So your nails are a special modification part of the external layer of your skin, which is kind of cool. Okay, where do we find nails? On the tips of our fingers and the tips of our toes. All right, those are the dorsal surfaces. All right, so they offer protection. How many times have you been grateful that you've had a nail, all right, and you drop something not too heavy, I hope, onto your fingernail or onto your toenail, and you're thinking, oh, if I didn't have a nail there, that would have hurt more. Well, they offer protection. They also offer help in gripping things. You ever get a splinter, all right, it helps to pull that splinter out with your fingernails. So let's talk about the anatomy of the fingernails. When you are sitting at home clipping your fingernails or toenails over the garbage, that part of the nail that you're clipping off is what we refer to as the free edge. So you're liberating and freeing that part of the nail, all right, from your body. So that is the free edge, and it's white because there's no blood vessels underneath, no capillaries, okay? Now when we start to move over the nail itself, all right, now we're in the area of the nail body, okay? When you look, it should be pink underneath, all right, and that's because of the capillaries. Now there is a test that you can do to kind of give to get an idea of how well, if someone's coming in and complaining of circulation issues. A nice test that you can tell to help determine that is you pinch or you press down on the top of their fingernail for like 5-10 seconds, then you release it. All right, it'll immediately be white, but then it should turn right back to pink. That should happen almost immediately. If it takes longer than a few seconds, 5 seconds or more, then they have a circulation problem. All right, it's called the capillary refill test. All right, and then the final part of the nail that we don't see is the nail root. Much like we don't see the root of your hair, all right, the nail root, this is the part of the nail that's embedded under or in the skin. I go to this picture here, okay, and kind of zoom in. Here's the free edge. This is the part we cut off, okay? And then here's the nail body. That's the majority of the nail that we see. And then underneath, all right, is the nail root. All right, but this whole hardened structure, that is referred to as the nail plate. This whole hardened structure, That includes the root, all right, the body, and the free edge. That is the nail plate. Okay. And it's the nail plate that lays on top of, all right, the epidermis there in your skin. That epidermis that it covers is the nail bed. All right, so I'll show you a picture. Here's the nail bed, this pinkish structure right down here. Okay, so the nail plate sits right on top of the nail bed. All right, a couple other things here. Quickly is the nail matrix. You remember the hair matrix? That was the area of the hair that, oh, and when I say you should remember that, I'm referring to lab. Sorry, I know I haven't covered this material in lecture, but in the lab for the identification there back in chapter 6, we talked about the nail matrix. And the nail matrix is that area of the hair that is actively, those cells are actively dividing to grow the hair. Well, that's what the nail matrix here is. All right, it's the area of the nail plate that's actively dividing, okay, that's growing the nail out, all right, from proximal to distal, okay. Now there's a whitish area on your nails, and you can look right where the nail kind of enters into the skin there. You'll notice this little white area, and we call that the lunula. And I think of lunar, for whatever reason, the moon, and at night the moon appears white. So this is this whitish area here that's at the proximal end of the nail. And you can see it here, it is right here. And again, that's a thickened area of the epidermis, so you can't see the pinkish color underneath it, all right, because it's a thickened area of the epidermis and the capillaries aren't showing through. All right, then on either side of the nail, what we call the nail folds, and that's just basically where the skin on the either side, the lateral portions of the nail, are covering the nail. All right, then the last two parts are the epiconium and the hypoconium. All right, the epiconium, all right, is what we also call the cuticle. All right, and this is the part of where the dermis actually, all right, attaches onto the nail bed itself, or nail plate, excuse me. Zoom in. All right, that's this little bit of skin or tissue right there that attaches onto the nail body or the nail plate. All right, and that's the epiconium or the cuticle. And then the last one is the hypoconium, and that's the part right here where the free nail edge hangs over the epidermis here, and it's kind of up here in this little region right there. All right, and that's the hypoconium. Okay, that's the part, all right, of the outer layer, your stratum corneum, that sits right underneath the free nail edge. All right, this one's going to be kind of a short one. All right, actually, I don't know how much time has passed, but this is going to be kind of a short video. I just wanted to get something out there. And then what I'm thinking about doing is I have some recordings from my other classes that I can share with you folks that has a little bit more material. Not that there's not a lot of material here, but I feel like, you know, if I give you some of these other videos here, it can go into a little bit more detail, and some of you might find that a little bit more helpful than the reviews. So I think I might just start using some of my other class. You'll hear some discussions going on in the class, obviously, but I think I'm going to do that because it'll give you a little bit more detail, and hopefully it'll be helpful. We'll still obviously have our Blackboard collaborate sessions. I'll be sending out announcements, obviously, but I think that we'll kind of go from there and see how that works out. All right. Obviously, if there's any questions, always email me. I try to respond as quickly as I can. And that being said, enjoy this video, keep studying, and the best of luck.